All right, let's go. Number 10. Yersinia pestis in the Neolithic. In 2018, a geneticist named Simon Rasmussen at the Technical University of Denmark published a paper in the journal Cell that quietly rewrote the history of the deadliest bacterium humanity has ever encountered. His team had been sifting through ancient DNA extracted from the teeth of Neolithic farmers buried in Sweden roughly 5,000 years ago. In a passage grave at a site called Frälsegården, they recovered genetic material from the molar of a young woman who had died around 3000 BC. What they found inside her tooth shouldn't have been there. Not at that depth in the timeline. Not in a body that predated every recorded plague in human history by 3 millennia. The bacterium was Yersinia pestis, plague. The strain has since been designated GOC2, and it is one of the oldest plague genomes ever sequenced from a human body. The young woman whose tooth carried it lived during the closing centuries of the European Neolithic, an era when farming communities across the continent had spent 2,000 years clearing forests, raising livestock, and building the first dense human settlements the world had ever seen. She was buried in a stone chamber alongside other members of her community. Her bones gave no indication of violence. Her skeleton offered no obvious cause of death. But the bacterium hiding in the pulp of her tooth was capable of killing a healthy adult in under a week. The implications of finding plague at that date are difficult to overstate. The Justinian pandemic of the 6th century AD and the Black Death of the 14th century have long defined the human imagination of mass mortality. Together they killed somewhere between 100 and 200 million people across recorded history. The GOC2 strain predates both events by more than 3,000 years. Plague is not a medieval invention. It is not a Bronze Age scourge. It was already circulating in human bodies before the first stone was lifted at Stonehenge. But here's where the discovery becomes genuinely disturbing. The timing of the GOC2 strain aligns almost precisely with one of the most poorly understood demographic events in European prehistory. Around 3000 BC, a cascade of abandonments swept across the Neolithic world. Farming communities that had occupied the same valleys for generations vanished from the archaeological record. Population estimates derived from radiocarbon-dated settlements show a continent-wide collapse. The grain stores empty. The longhouses fall silent. The pollen records show forests reclaiming fields that had been worked for centuries. Something hollowed out the European Neolithic, and no one has ever fully explained what. Rasmussen's team proposed a mechanism. The same centuries that produced the GOC2 strain also produced the first true megasettlements in human history. At Trypillia, in modern Ukraine, archaeologists have mapped urban centers housing as many as 15,000 people, arranged in concentric rings of timber houses spanning hundreds of hectares. These were the largest human gatherings the planet had ever sustained. They were also the first laboratories in which a respiratory pathogen could find enough hosts to evolve, persist, and travel. Trade routes radiated outward from these centers, carrying flint, copper, amber, livestock, and almost certainly the microorganisms that lived inside human lungs and bloodstreams. The GOC2 genome lacks some of the genetic adaptations that would later make plague flea-borne. Its mode of transmission appears to have been respiratory, person to person, breath to breath. In a settlement of 15,000, a pneumonic pathogen needs no rats. It only needs neighbors. We will likely never know how many died. Prehistoric demography leaves no obituaries, no chroniclers, no parish registers. What it leaves is silence in the soil, settlements that simply stop appearing, burial chambers sealed and forgotten. A young woman in a Swedish passage grave with a bacterium in her tooth that would, 4,500 years later, kill a third of Europe under a different name. The first Great Plague did not arrive in the 6th century. It was already moving through human bodies when the megaliths were new, when the pyramids had not yet been imagined, when the people dying of it had no word for what was killing them. Number 9. Tuberculosis beneath the sea. In 2008, microbiologists Helen Donahue and Mark Spiegelman published a paper in PLOS One describing genetic material extracted from 2 skeletons that should not have survived to be analyzed at all. The remains belonged to a mother and her infant. They had been dead for approximately 9,000 years, and they had been lying together beneath the Mediterranean Sea, 8 to 12 meters down, off the coast of Mount Carmel in modern Israel, for nearly all of that time. The site is called Atlit Yam. It is a Neolithic village, fully submerged, preserved in the dark, quiet sediment of the seafloor. Stone hearths, wells, ritual installations, the foundations of houses, all of it sitting beneath the waves, where Bronze Age sailors would later pass overhead without knowing what lay beneath their hulls. The village was swallowed at the end of the last glacial period, when meltwater from the great northern ice sheets raised sea levels and pushed the Mediterranean inland over coastlines that had been dry for millennia. The people of Atlit Yam did not see the catastrophe coming in a single afternoon. They watched the water creep up, year after year, until their homes were no longer their homes. But the water was not the only thing killing them. When Donahue and Spiegelman analyzed bone fragments from the mother and infant, they recovered DNA from Mycobacterium tuberculosis. The genetic signature was unmistakable. Tuberculosis, the same disease that would later devastate medieval cities, hollow out 19th century slums, and kill more human beings across history than any other single pathogen. Here it was, already at work, in the bones of a Neolithic woman cradling her child on a coastline that no longer exists. The dates make the discovery extraordinary. 9,000 years, among the oldest confirmed cases of human tuberculosis ever recovered, older than the pyramids by more than 4,000 years, older than writing, older than the wheel, older than nearly every cultural marker we use to define the beginning of civilization. The disease was already inside us. It had already learned how to live in human lungs, how to pass between mother and child, how to wait quietly in the bone marrow for years before announcing itself. But here's where the implication becomes genuinely unsettling. Atlit Yam was not a band of nomadic hunters passing through. It was a settled village. Its inhabitants had begun domesticating cattle. They had committed to a way of life that placed them in close, daily, breathing contact with large mammals for the first time in human evolutionary history. And tuberculosis is a zoonotic disease, a pathogen that crosses from animal hosts into human ones, then adapts, then never leaves. The cattle that fed Atlit Yam may also have killed it. The same innovation that allowed Neolithic communities to grow beyond the carrying capacity of foraging, the same revolution that built the foundation of every civilization that would follow, opened a biological door that could never be closed again. The mother and infant are evidence of that door swinging open. They are among the earliest known casualties of a transaction the human species made 9,000 years ago and has been paying for ever since. Every tubercular death in every century afterward, the poets coughing blood in Roman villas, the children in Victorian tenements, the millions still dying in the modern world, traces back through an unbroken chain to coastlines like the one off Mount Carmel, where someone first decided to keep cattle close. The bones still rest in the sediment. The village still sits beneath the Mediterranean, its hearths cold, its wells flooded, its dead preserved in the silence of the deep shelf. And in the genetic record of those two skeletons lies a warning the Neolithic could not read. The disease they carried was learning. It was getting better at us, and it would follow our descendants into every settlement they ever built. Number 8. Leprosy in Bronze Age India. In 2009, bioarchaeologist Gwen Robbins-Shug at Appalachian State University published a paper in PLOS 1 describing a skeleton that pushed the documented history of one of humanity's most feared diseases back by more than 1,000 years. The remains had been excavated at Balathal, a Chalcolithic settlement in Rajasthan, and dated to approximately 2000 BC. The bones belonged to a middle-aged man. The lesions on his skull, his nasal aperture, and his extremities matched a pattern that physicians have recognized for millennia. Mycobacterium leprae had been at work on his body for years before he died. This was the oldest confirmed case of leprosy outside Africa. The implications rippled outward immediately. If the disease had reached interior Rajasthan by 2000 BC, it had been traveling far longer than the historical record suggested, carried along the trade corridors that knit Africa, the Middle East, and South Asia into a single biological theater. The cargo moving between these regions wasn't only copper, lapis lazuli, and grain. It was bacteria. The same routes that built civilizations were the routes that infected them. The burial itself was unusual, in ways that suggest his community knew exactly what he had become. He had been interred inside a stone enclosure, his body covered in ash. This was not the standard treatment for a Chalcolithic dead man in Rajasthan. Ash carried ritual weight across the ancient subcontinent. Purification, separation, transformation. The stone enclosure isolated him from the surrounding earth. Whatever his neighbors believed about the lesions consuming his face and limbs, they buried him as something requiring containment. The grave was a quarantine that outlasted him. What makes this skeleton particularly disturbing is its timing. The man at Balathal died during the mature Harappan phase of the Indus Valley Civilization, the high tide of urban achievement in Bronze Age South Asia. Mohenjo-Daro and Harappa, the great cities of the Indus Plain, housed tens of thousands of inhabitants each. Standardized brick, gridded streets, municipal drainage systems, granaries, public baths. This was one of the three cradles of human civilization, an urban culture spanning more than 1 million square kilometers, sustained by a population estimated at 5 million people, moving along the same trade arteries that were now moving Mycobacterium leprae between continents. Within a century of the Balathal burial, those cities began to fail. By 1900 BC, the great urban centers of the Indus were depopulating. The standardized weights stopped circulating. The script went silent. The drainage systems clogged with debris that no municipal authority cleared. The population dispersed eastward and southward into smaller agricultural settlements, abandoning the urban project entirely. Climate shifts, river course changes, and the weakening of the monsoon are usually invoked to explain the collapse. They are almost certainly part of the story, but they are not the whole of it. A leprous skeleton sealed in ash at the periphery of the civilization, dated to its final flourishing, opens a quieter possibility. Dense urban populations, connected by long-distance trade to disease reservoirs across 2 continents, were doing for the first time in human history what cities have done ever since. They were incubating pathogens. They were concentrating susceptible hosts. They were importing infections faster than immune systems could adapt. Leprosy is slow. It does not empty a city in a season, but it is one signal of a microbial environment that had grown unfamiliar, and the man at Balathal is the signal we can still read. How many other diseases moved through those streets unrecorded, leaving no lesions on bone, no diagnostic scars for a bioarchaeologist to identify 4,000 years later, is a question the Indus script may have answered before it fell silent. The bones at Balathal are what remains. The cities they belong to are dust. Number 7. Helicobacter in the Iceman. In September 1991, 2 German hikers crossing the Ötztal Alps along the Austria-Italy border noticed something dark emerging from the melting ice of a high mountain pass. They assumed it was a recent climbing fatality. It was not. The body locked in the glacier had been lying there for approximately 5,300 years, preserved by cold and altitude into something no archaeologist had ever seen before. A complete prehistoric human. Skin, organs, stomach contents, tattoos, tools, clothing. An entire Copper Age individual, delivered intact into the late 20th century. He was named Ötzi, and his body, more than any text or ruin or buried artifact, became the most detailed single record of a human life from the period before writing existed. For 2 decades, researchers cataloged his anatomy, his last meals, the arrow wound in his shoulder that killed him, the copper axe at his side, the 61 tattoos mapped across his joints, the wear patterns on his teeth, the pollen in his lungs that pinpointed the season of his death. Ötzi was not a civilization. He was one man. But one man preserved this completely, is a kind of archive, and the deeper researchers looked, the more passengers they found riding inside him. In 2016, a team led by Frank Meixner at Urac Research in Bolzano published a paper in Science describing what they had recovered from the contents of his stomach. Not food, not parasites, a bacterium, Helicobacter pylori, the same organism that infects roughly half of all humans alive today, the cause of ulcers and stomach cancer in modern populations, was still present in his gut after 5 millennia in the ice. They sequenced it. They reconstructed its genome. They placed it on the evolutionary tree of every H. pylori strain ever cataloged, and it did not belong where he died. Modern Europeans carry a hybrid strain of H. pylori, a genetic blend produced by the mixing of African and Asian lineages over thousands of years of human migration. Ötzi's strain was not hybrid. It was almost purely the ancestral Asian type, a virulent lineage now found primarily in South and Central Asia. The bacterium living in the stomach of a Copper Age man who died in the Alps was the same bacterium currently colonizing populations from Pakistan to northern India. The European hybrid had not yet formed. The migrations that would produce it had not yet finished. Ötzi's microbiome was a snapshot of a world before that mixing was complete. The implications cut in several directions at once. It meant that the human populations who eventually became modern Europeans were still being assembled 5,300 years ago at the bacterial level. It meant that the diseases carried by those populations were also still being assembled, and it meant that a single frozen body could rewrite the timeline of how pathogens traveled with humans across the continents. H. pylori was not the only passenger. Researchers also found genetic traces of Borrelia, the spirochete responsible for Lyme disease, making Ötzi the oldest known human case. His intestines contained the eggs of whipworms, parasitic nematodes that would have caused chronic abdominal distress throughout his life. He carried, simultaneously, a stomach pathogen, a tick-borne infection, and an intestinal parasite. He was, by any modern clinical standard, a sick man walking through high country with an arrow wound he would not survive. What makes Ötzi particularly unsettling is the implied scale. He is one preserved body out of millions who lived and died in his era. If a single individual carried this much microbial weight, the populations he came from carried it too. The villages, the trade routes, the seasonal camps, all of them moving pathogens through bodies that had no concept of contagion and no defense against what was already inside them. The ice kept his record. Everyone else's dissolved. Number 6. Malaria and the pharaohs. In 2010, a team led by Egyptologist Zahi Hawass and geneticist Karsten Pusch published findings in the Journal of the American Medical Association that rewrote what we thought we knew about the death of the most famous boy in human history. Tutankhamun, who died around 1323 BC at approximately 19 years of age, had been examined, x-rayed, scanned, and theorized over for nearly a century. Murder, chariot accident, infected leg wound, genetic disorder. Every hypothesis had been proposed and defended. What the DNA analysis revealed was simpler and more terrible than any of them. Plasmodium falciparum, the deadliest strain of malaria parasite known to medicine, recovered from the desiccated tissue of a pharaoh who had been dead for over 3,000 years. The boy king had malaria when he died. Not a single mild bout. Multiple infections of the most lethal variant in the parasite family. And he was not the first. Pre-dynastic Egyptian mummies, individuals who lived before the unification of Upper and Lower Egypt, before the First Dynasty, before the very concept of pharaonic civilization existed, have yielded the same parasitic DNA. Malaria was not a late arrival in Egypt. It was already there when Egypt began. The disease and the civilization grew up together along the same river. The Nile made this inevitable. The annual flooding that turned the desert into the most productive agricultural land in the ancient world also created something else. A continuous belt of standing water, irrigation canals, and slow-moving channels, stretching for over 1,000 kilometers. A perfect, permanent breeding habitat for the Anopheles mosquito. The same hydrology that fed the granaries of Memphis and Thebes incubated the parasite that would kill their pharaohs. Every harvest came with a hatching. Every flood season delivered grain and fevers in the same water. Egyptian medical papyri described the symptoms with clinical precision: cyclical fevers, chills that returned every few days, enlarged spleens, the pallor that physicians of the New Kingdom recognized but could not explain. They had no concept of a parasite. They had no concept of a mosquito as a vector. They had only the pattern of the disease itself, watched and recorded across generations of scribes, who knew it as intimately as they knew the rise of Sirius. They named it, they treated it, they buried its victims, and they could do nothing to stop it. But here is where the implication extends beyond a single dynasty. Malaria is estimated to have killed hundreds of millions of human beings across the span of our history. More than any war, more than any other single pathogen we can name, it has shaped genomes. In the populations surrounding the Nile, in sub-Saharan Africa, in every region where Plasmodium falciparum became endemic, human DNA itself began to bend under the pressure. Sickle cell trait, thalassemia, glucose 6-phosphate dehydrogenase deficiency, adaptations that carry their own lethal costs were selected for, because they offered partial protection against a parasite that was killing children before they could reproduce. The disease was rewriting the species. Consider what this means for Egypt, the civilization that built the pyramids, that codified writing, that maintained centralized authority for 3,000 years, did so while burying an unknowable percentage of its population to a fever it could not understand. The architects of Giza, the engineers of the Karnak temple complex, the scribes who copied the Book of the Dead, all of them lived inside a landscape where infants died in cycles dictated by mosquito hatchings. The pharaoh on the throne carried the same parasite as the laborer in the field. Tutankhamun's gold death mask and his plasmodium DNA were buried in the same coffin. The disease did not end Egypt. Egypt simply learned to absorb its losses, generation after generation, and to call that absorption normal. It would not be the only ancient society to do so. The pattern of a pathogen embedded so deeply into a civilization that the civilization itself becomes a vector. That pattern would repeat, on a scale that dwarfs the Nile, in the Bronze Age world that surrounded it. Number 5. Hepatitis B across the Bronze Age. In 2018, a team of geneticists led by Eske Willerslev at the University of Copenhagen published a paper in Nature that quietly reframed what we know about the invisible passengers riding inside ancient human populations. The team had extracted viral DNA from 25 Bronze Age and Iron Age skeletons recovered from burial sites stretching from Germany to the Eurasian steppe. What they found wasn't a single localized outbreak. It was the same virus, hepatitis B, persisting across thousands of kilometers and thousands of years inside the bodies of people who never knew they carried it. The oldest sample came from a Yamnaya burial, dating to approximately 4,500 years ago. The Yamnaya were not a marginal population. They were one of the most consequential cultures in European prehistory, horse-riding pastoralists from the Pontic-Caspian steppe, whose Bronze Age migrations reshaped the genetic foundation of Europe so thoroughly that nearly every modern European carries fragments of their ancestry. They brought language, they brought horses, they brought metallurgy, they brought new burial practices, new social structures, new gods. They also brought hepatitis B. The virus appears in skeletons separated by enormous distances. Germany, the Carpathian Basin, the steppe corridors east of the Volga. Different individuals, different generations, different communities, all carrying genetic traces of the same pathogen. The geographic spread mirrors almost exactly the migration routes that geneticists have independently mapped through human DNA. Wherever the Yamnaya expansion reached, the virus reached with it. The people moved, the virus moved inside them. The numbers are worth sitting with. 25 ancient skeletons might sound modest, until you consider what recovering viral DNA from 4,500-year-old bone actually requires. Hepatitis B is a tiny, fragile virus. Its genetic material degrades quickly. To recover it from skeletons buried for over 4 millennia means the original infection load must have been enormous, and the prevalence across the population must have been high enough that random burials across an entire continent keep turning up positive. The 25 samples are not the disease. They are what remains of it. The actual infected population during the Bronze Age may have numbered in the millions. But here's where the discovery becomes genuinely disturbing. Several of the viral genomes recovered from these skeletons belong to genotypes that no longer exist, extinct lineages of hepatitis B. Strains that infected Bronze Age humans across Eurasia persisted for generations, traveled with migrating populations, and then vanished from the modern human virome entirely. Something erased them, whether through host extinction, viral competition, or shifts in human population structure that broke their transmission chains. Entire branches of the hepatitis B family tree died with the people who carried them. The virus we know today is a survivor. It is not the whole story. It is the fragment of a much larger viral world that moved through Bronze Age Eurasia and then disappeared. What makes the hepatitis B finding particularly unsettling is the nature of the disease itself. This is not a pathogen that announces its presence. Acute infection can be mild or asymptomatic. Chronic infection settles into the liver and works silently for decades, scarring tissue, triggering failure, eventually producing cancer. A Bronze Age herder infected at 20 would not have known. His community would not have known. He would have ridden, traded, married, fathered children, and died at 40 of what his people would have understood as simple weakness or wasting. No epidemic, no visible plague, no mass graves, no archaeological signature at all. Just steady, generational attrition, invisible to every method we use to read the past. This is the part that should make the hair rise on the back of the neck. The 4.2 kiloyear event left dust in marine cores. The Toba eruption left ash on 3 continents. The Stoega slide left sand deposits 80 kilometers inland. Catastrophes write themselves into the rock. Hepatitis B writes nothing. A pandemic operating at the scale of an entire continent for thousands of years would leave no detectable trace in any conventional archaeological record. No destruction layer. No abandonment horizon. No shift in pottery styles or burial practices. The bones look ordinary. The settlements look ordinary. The story looks ordinary. Only the DNA betrays it. And the DNA only betrayed it because Willerslev's team thought to look. Until 2018, the conventional understanding was that hepatitis B was a relatively recent companion of human civilization, a few thousand years old at most. The Bronze Age samples pushed its documented history back into the heart of the migrations that built modern Europe. The implication is that the Yamnaya did not simply replace the Neolithic farmers of the European interior through demographic expansion. They may have arrived carrying viral loads against which those farmers had no immunity. Whether hepatitis B contributed materially to the population turnover or merely rode along as a passenger, it was present. It was widespread. It was killing people slowly across an entire continent during the formative centuries of European prehistory. The truly disturbing aspect is what this single discovery implies about everything else. If hepatitis B can hide in the Bronze Age record for over 4,000 years, waiting for sequencing technology sophisticated enough to find it, how many other viruses are still hiding? How many silent pandemics moved through prehistoric populations, leaving no archaeological signature, no cultural memory, no record of any kind, simply waiting in fragments of degraded DNA inside bones we have already excavated and catalogued and shelved? The answer is unknowable. The Bronze Age skeletons in museum collections across Europe may contain the genetic ghosts of a dozen lost pandemics we have not yet thought to look for. The virus moved with the people. The people built the world we inherited. And the diseases they carried into that world were already older than any civilization that would ever exist. Number 4. Parvovirus in the Stone Age. In 1975, virologist Yvonne Cossart at a London laboratory was screening blood samples for hepatitis B when she noticed an anomaly in sample number 19 of panel B. What she had stumbled onto wasn't hepatitis. It was an entirely new human virus, a tiny single-stranded DNA pathogen that would come to be called Parvovirus B19. The naming was arbitrary, drawn from the position on her sample tray. The virus itself was anything but arbitrary. It would turn out to be one of the most widespread infectious agents on Earth, present in nearly every modern human population, transmitted by something as casual as a cough across a room. What no one in 1975 could have known was that the virus had been doing exactly this for at least 7,000 years. In recent ancient DNA studies of Neolithic and Mesolithic European remains, researchers recovered intact fragments of Parvovirus B19 genetic material from skeletons buried in the soil for 7 millennia. The recovery is remarkable for a reason that is easy to underestimate. Parvovirus is fragile. It is a small, naked virus with no protective envelope, the kind of pathogen that should not, by any reasonable expectation, leave a readable signature in bone after 7,000 years. And yet it did. The genetic record of the disease survived in the marrow of people who died before the wheel, before writing, before metallurgy. They carried it. We can still read it. The numbers around what Parvovirus B19 actually does to a human body explain why its presence in the Neolithic record is so disturbing. In a modern healthy adult, the infection often passes as a mild rash and a few days of joint pain. In a pregnant woman, the same virus can cross the placenta and shut down fetal blood production, causing miscarriage or stillbirth. In an immunocompromised individual, it can trigger sudden severe anemia by destroying the precursor cells in bone marrow that produce red blood cells. In a child with an existing blood disorder, it can be lethal within days. The virus targets the very machinery that keeps a body alive. Now place that virus inside the Neolithic transition, around the same window in which Parvovirus B19 appears in the ancient DNA record. Human beings were doing something unprecedented. They were settling down. They were clustering into permanent villages. They were sharing enclosed structures with their livestock, their grain stores, and each other in densities the species had never sustained before. Parvovirus B19 spreads through respiratory droplets. It requires bodies close enough to share the same air for long enough to inhale what the sick are exhaling. Mobile foraging bands of 20 or 30 individuals do not sustain that kind of transmission. Sedentary farming villages of 200 do. The implication is precise and unsettling. The disease did not merely accompany the Neolithic transition. The Neolithic transition was the condition that made the disease possible at the scale we now find it. Agriculture didn't just feed humanity. It incubated humanity's pathogens. And the targets of that incubation were the most vulnerable members of the new settled communities. Pregnant women losing pregnancies they did not understand they were losing. Infants dying of anemia in the arms of mothers who had no concept of red blood cells. Children with inherited blood disorders that we now recognize and treat, collapsing without warning. None of it would have registered as an epidemic. It would have registered as bad luck, as the work of spirits, as the price of living where you lived. 7,000 years later, the same virus circulates in nearly every population on the planet. It is in the schools. It is in the hospitals. It is in the air of any crowded room. The Neolithic villagers who first sustained its spread are gone. The virus they unknowingly cultivated is still here, still spreading, still doing exactly what it learned to do when human beings first decided to stop moving. Number 3. Salmonella and the Cocoliztli. In 2018, a team led by Åshild Vågene at the Max Planck Institute for the Science of Human History published a paper in Nature Ecology and Evolution that identified the genetic signature of a single bacterium recovered from the teeth of skeletons buried at Tepozcalula Yucunda in Oaxaca. The bacterium was Salmonella enterica paratyphi C. The skeletons belonged to victims of one of the most catastrophic epidemics ever documented in human history, and until that paper, no one had been able to name what killed them. The Aztecs had a word for it, Cocoliztli. In Nahuatl, it simply means pestilence, but the word, when it appears in colonial-era manuscripts, carries the weight of something the survivors could not describe in any other terms. The epidemic that swept through central Mexico between 1545 and 1548 killed an estimated 80% of the population. A second outbreak in 1576 killed another 2 million. Pre-contact Mesoamerica had supported a population estimated between 15 and 30 million people. Within a century of European arrival, that population had been reduced by over 90%. The numbers are not casualties of war. They are not the slow attrition of conquest. They are the signature of biological catastrophe operating at a scale that has no modern parallel. The Spanish physician, Francisco Hernandez, witnessed the 1576 outbreak and recorded what he saw. High fever, a tongue that turned black, bleeding from the eyes and the nose, death within days of the first symptoms. Hernandez was a trained observer, court physician to Philip II, sent to the New World specifically to catalog its medical knowledge. He had no framework for what he was watching. The disease did not match smallpox. It did not match measles. It did not match typhus or anything in the European medical literature. Whatever was killing the people of central Mexico was something the Old World had no name for. For nearly 5 centuries, the identity of Cocoliztli remained one of the great unsolved questions of epidemiology. Then Vågene's team extracted ancient DNA from the dental pulp of 10 individuals buried in the cemetery at Tepozcalula Yucunda, a Mixtec town that had been abandoned almost immediately after the 1545 outbreak. The DNA preserved in those teeth carried fragments of a pathogen that should not have been there. Salmonella enterica paratyphi C is a bacterium that causes enteric fever, a systemic infection that today exists at low levels across parts of Africa and Asia. In the 16th century, introduced to a population with no prior exposure into a landscape already destabilized by conquest, famine, and forced labor, it became something else entirely. It became an extinction event for a civilization. But here's where the story becomes relevant to everything that came before it on this list. The Cocoliztli epidemics are not prehistoric. They are documented. They have eyewitnesses, named physicians, colonial records, parish death registers. We know what happened because Spanish bureaucrats counted the dead, and because the Mixtec abandoned a town so completely that its dental remains lay undisturbed for the molecular biologists who would eventually arrive to read them. Cocoliztli is the rare case where the bacterium, the bodies, and the written record all survive together. And what it shows is this: a single bacterium, introduced into a population without immunity, can collapse a civilization of tens of millions within a generation. Not weaken it, not diminish it, erase it. The city's empty, the agricultural systems fail, the languages contract, the temples are repurposed by the survivors of a different culture entirely. Within decades, the world that existed before the bacterium arrived becomes a world that can only be reconstructed from ruins and the careful work of strangers reading bones. Now consider what the prehistoric record cannot show us: the communities that vanished before writing existed, the settlements that emptied for reasons no chronicler recorded, the genetic bottlenecks that appear in our DNA without explanation. Cocoliztli is the window. It is the one event we can see clearly, because the timing was late enough for ink and early enough for the bacterium to leave its name in a tooth. Everything before it, every prehistoric collapse, every abandoned site, every population crash written into ancient genomes, happened in the silence on the other side of that window. Number 2. Smallpox and the Viking world. In 2020, a team led by geneticist Eski Willerslev and virologist Barbara Meuleman published findings in Science that rewrote what we thought we knew about one of humanity's oldest killers. Working with samples drawn from 11 Viking-era skeletons scattered across northern Europe, the team recovered fragments of variola virus DNA. The bones came from Sweden, Denmark, Norway, the United Kingdom, and Russia. The dates ranged from roughly 600 to 1050 AD, and the virus they extracted from those long-buried bodies was not the smallpox that walked into the 20th century. It was something older, something that no longer exists. The strains recovered from the Viking dead belonged to an extinct sister clade of modern smallpox, a branch of the variola family tree that diverged from the lineage humanity would eventually defeat in 1980, splintered off into its own evolutionary trajectory, and then vanished. The virus that killed those Vikings is gone. The virus that killed an estimated 300 million people in the 20th century alone descended from a different branch of the same dark genus. Two parallel killers, both circulating, both shaping human populations, only one of which survived long enough to be eradicated. The implications cut in several directions at once. For most of the 20th century, the assumption among virologists was that smallpox emerged as a major human pathogen sometime within the last 2,000 to 3,000 years. Textual evidence and the famous lesions on the mummy of Egyptian pharaoh Ramesses V, who died around 1145 BC, suggested variola had been killing humans for at least 3 millennia. But the Viking samples push the story into a different shape entirely. They demonstrate that smallpox was already a diversified, geographically dispersed pathogen by the 7th century AD. Genetic clock analyses anchored to those samples suggest the virus's origin may stretch back at least 4,000 years, evolving alongside human populations across vast stretches of prehistoric time we have no written record of. What this means is straightforward and terrible. Smallpox was killing humans long before any civilization existed to document its passage. The disease that would later annihilate empires, that would arrive in the Americas in the 16th century and reduce indigenous populations by as much as 90% in some regions, that would shape European demography for over 1,000 years, had already been circulating among prehistoric communities for millennia. It killed kings whose names we will never know. It emptied villages whose locations we will never find. It traveled along trade routes and migration corridors that left no archaeological trace. The Viking samples are simply the oldest specimens we have managed to recover. They are not the beginning of the story. They are a single horizontal slice through a much longer history of contagion that humanity lived through without writing any of it down. Consider what the geographic spread alone reveals. 11 skeletons across 5 modern countries, all carrying related but distinct strains of an extinct variola lineage, all dating to a window of roughly 450 years. This was not a localized outbreak. This was a continent-wide presence of a virus that had already differentiated into multiple subtypes, suggesting it had been moving through northern European populations for centuries before the earliest Viking samples were even buried. The trade networks that carried amber and silver also carried infection. The long ships that crossed the North Sea carried sailors whose bodies were hosts. And the extinct sister clade itself raises a question the researchers could not answer. Why did one branch of variola survive into modern history while the other disappeared? Was the Viking-era strain less transmissible, outcompeted by its more virulent cousin? Did it burn through its host populations too quickly to sustain itself? Did some shift in human density, in trade patterns, in climate, simply remove the conditions it needed to persist? We do not know. What we know is that smallpox is older than any civilization that ever feared it. We know that for thousands of years before the Greeks, before the Egyptians built their final pyramids, before any culture developed the writing systems necessary to record an epidemic, variola was already moving through human bodies, killing in patterns we cannot reconstruct. The Viking dead are not evidence of smallpox's arrival in Europe. They are evidence that it had been there, in some form, for so long that we may never trace its true beginning. The virus that defined modern history was an old killer wearing a new face. Number 1. Treponematosis in the Americas. In the 1980s, paleopathologist Bruce Rothschild began surveying skeletal collections across North and South America, looking for a very specific signature. Not fractures, not arthritis. A particular pattern of pitting and remodeling on long bones, on skulls, on tibia, the kind of damage that only one family of bacteria leaves on human remains. He found it, not in a single graveyard, not in a single region, across the entire hemisphere, in skeletons dated 6,000 years old and older. The lesions were unmistakable, and they were everywhere. The bacterium responsible is called Treponema pallidum, and it is one of the strangest pathogens in human history. It causes 4 distinct diseases depending on the subspecies: syphilis, the sexually transmitted form; yaws, a tropical skin disease; begel, a non-venereal infection spread by contact; and pinta, a disfiguring condition that attacks pigmentation. The bacteria are so similar that distinguishing them in ancient bone is one of the most difficult problems in paleopathology. But the bones tell us something the historical record never did. Treponemal disease was circulating in the Americas for thousands of years before any Europeans set foot on the continents. The numbers establish the timeline: 6,000 years, possibly longer. Generations layered upon generations of indigenous American populations carrying a pathogen that European medicine had no concept of. No name for it, no treatment, no immunity. The disease was not waiting to be discovered. It was waiting to be exported. In 1493, Columbus's ships returned to Spain. In 1495, during the French invasion of Naples, something erupted in the city that no European physician had ever seen. Soldiers developed sores. The sores spread, faces collapsed, bones rotted. The disease moved through armies and brothels and royal courts with a speed that terrified every medical authority on the continent. They called it the French disease. The French called it the Neapolitan disease. The Polish called it the German disease. Nobody wanted to claim it. Within 5 years, it had reached every corner of Europe. Within a generation, an estimated 5 million Europeans were dead. The Columbian exchange hypothesis proposes that Columbus's returning sailors carried treponemal disease back across the Atlantic, and that the strain that emerged in Naples was the American pathogen meeting a European population with no evolutionary preparation for it. The timing is suggestive. The first documented European epidemic begins 24 months after Columbus's return. The geography is suggestive. Naples in 1495 was full of Spanish mercenaries who had served in the New World expeditions. The skeletal record is suggestive. European bones from before 1493 show almost no convincing treponemal lesions. After 1495, the lesions are everywhere. But here's where the story becomes genuinely vertiginous. Ancient DNA work by Verena Schünemann at the University of Zurich has now identified treponemal lineages preserved in pre-contact Brazilian remains. The genetic material is real. The dating is secure. The diseases existed in the Americas, in identifiable molecular form, centuries before Columbus's ships left harbor. Whatever circulated through indigenous populations for those thousands of years was not a single pathogen, but a family of them, evolving, branching, adapting to human hosts in ways that European medicine would spend the next 500 years failing to fully understand. The implications are staggering. a disease that emerged among prehistoric American peoples, whose names we do not know, whose languages we cannot reconstruct, whose civilizations left no written record, reached across an ocean and killed 5 million Europeans within decades of contact. The pathogen did not need conquistadors or treasure fleets or trade routes that took centuries to establish. It needed a single voyage. The microbe moved faster than empire, faster than commerce, faster than any human institution. By the time the survivors in Naples understood they were dying of something new, the disease had already rewritten the demographic future of an entire continent. And the indigenous Americans who had carried these treponema lineages for 6,000 years left no chronicle of their own suffering. We know they were sick because their bones tell us. We know nothing of how they understood the disease, how they treated it, how they incorporated it into their cosmologies. The lesions Rothschild documented are the only testimony those communities ever gave. A child in coastal Brazil 4,000 years ago, dying slowly of an infection that would one day cross the Atlantic and end the lives of millions of Europeans, left no name behind, only the bone. This is the pattern this list has been circling. Disease has been moving through human populations since before any human population could write the word disease. Pathogens evolved with us in the deep past, traveled with us, killed us in numbers we cannot reconstruct, and shaped the demographic foundations of every civilization that ever emerged from the prehistoric world. The Columbian Exchange was not the first time a disease from one continent reshaped the destiny of another. It was simply the first time someone wrote it down. Everything before that, every plague that swept through a Neolithic village, every infection that hollowed out a Bronze Age trade network, every epidemic that emptied a settlement we have not yet excavated, happened in silence. The victims left bones, they left lesions, they left genetic fragments that modern sequencers are only now beginning to read. They did not leave words. The prehistoric world was not killed by the things its inhabitants feared. It was killed by the things they could not see. The pattern, once you see it, refuses to be unseen. Across every entry in this countdown, the same silence repeats. The victims of prehistoric disease left no chronicles, no inscriptions, no funeral orations, no survivor accounts scratched into clay or stone. The communities that bacteria and viruses dismantled in the deep past were almost entirely preliterate, and the pathogens that ended them moved through populations that had no way to name what was happening, let alone record it for the future. For most of human history, our greatest killers operated in total documentary darkness. What changed is recent. Since the early 2000s, the ancient DNA revolution pioneered in Svante Pääbo's laboratory and extended by the generation of researchers who followed, has transformed bone fragments and tooth pulp into archives. Yersinia pestis pulled from a 5,000-year-old jaw, Hepatitis B reconstructed from Bronze Age skeletons, Tuberculosis read from the ribs of farmers who died before the first city was built. The dead are finally speaking, molecule by molecule, in a language we only just learned to hear. The plagues that erased prehistoric worlds are still inside the bones, waiting to be sequenced. If you want to see more videos like this, click the video on screen now